Hi, my name is Alecxyia Valdez, and for today's result presentation, I will be discussing the bacteria called Bacillobacter sylvestris. The environment where this bacteria was collected was in a sandy subtropical soil. Scientists isolated that soil sample in Namibia, which is a country in Africa. The temperature in which this bacteria grows is between 12 to 40 degrees Celsius, in other words, 54 to 104 degrees Fahrenheit. However, the best growth of this bacteria occurs when the temperature is between 25.5 and 31.1 degrees Celsius. If converted to Fahrenheit, it would be 78 to 88 degrees. The pH of this bacteria ranges from 4.7 to 9 on the pH scale. The bacteria can vary in terms of being acidic and slightly basic. This is because 4.7 is below the neutral line of 7, which makes it acidic, and 9 is above it, which makes it slightly basic. The bacteria does contain salt. The salt that is present is sodium chloride, also known as table salt, and this salt is available at our homes today, and it contains 0 to 1% of it. Some things that are special about this bacteria is that it is not mobile, meaning it doesn't move, and it's a chemoorganoheterotroph. This is unique because it falls into the same nutrition category as us humans. We both get our energy and carbon source from organic compounds like nucleic acids, fats, sugars, and proteins, which can only be obtained through food, which means that these microbes also eat food to obtain their carbon source and energy source. In addition to this bacteria is an aerobe, meaning it needs oxygen to grow. During my research of this bacteria, there was scarce information regarding soil relationships, so I had to think more broadly. I found out that this certain bacteria falls under a broader group called Acidobacteria. Acidobacteria is a predominant phylum in tropical agricultural soils. The role of an acidobacteria is to recover soils, participate in both carbon and nitrogen cycles, uptake ions and metabolism, and decompose matter. In terms of recovering soils, acidobacteria like this one are beneficial to a plant's soil nutrient cycling and plant growth. The role of this bacteria in the carbon cycle is to degrade complex polymers and oxidize carbon monoxide. This makes sense considering the fact that carbon monoxide oxidation results in a drier soil, which is the same as which it was taken from, which was a sandy subtropical one. The role it has in the nitrogen cycle is to reduce nitrate, nitrite, and nitric oxide. Reducing these will generate a greenhouse gas of nitrous oxide. This is generated by this type of microbe and has the role of altering the climate. Bacillobacter sylvestris grows best in warm weather. I can infer that colonies will be high in number in warm temperatures. The microbe can survive in different climates because of the nitrous oxide that they produce. Although the soil doesn't contain as much moisture as other soils, the microbes living in this environment have adapted to this and have the ability to survive and thrive in a sandy one. The information correlates to the environment because it was found in a subtropical climate. In addition to this, the carbon monoxide oxidation has contributed in making the soil more dry, which makes sense because the sample was taken from a dry environment, almost desert-like. Also, the name of this bacteria hints to where it was originated from. The name Bacillobacter tells us it was taken from Namibia because there is a root word of Nambi, which is similar to Namibia. Finally, I would discuss some of the research about this bacteria. Scientists found a widespread acidobacteria called Bacillobacter sylvestris and harvested it from a semi-arid savanna in Namibia. It is also a relatively new discovery as well. All documentation and research was complete in 2016. The soil sample was collected at the village of Mashari, located in Africa. In addition, there have been other research projects surrounding acidobacteria generally. One study was on micropopulations in sugarcane soil. It contained an abundant amount of acidobacteria groups. This concludes my part of the presentation, and I will now pass it on to the next presenter.
Hey, I'm Lachlan, and I'm going to be giving this portion of the presentation on Aquahabitans deichengensis. This bacteria resides in fresh water, and it thrives in warm temperatures. It can survive in environments ranging from 15 to 37 degrees Celsius. However, it thrives at the optimal temperature of 30 degrees Celsius, which is the equivalent of about 86 degrees Fahrenheit. Further, the optimal pH on the pH scale for this organism is 7, which means that it's neutral, neither acidic nor basic. Another interesting fact about them is that they can't survive in the presence of salt, namely sodium chloride or table salt. This is why they're only found in freshwater environments. Something unique about this bacteria is the fact that the study which made note of this specific strain's existence described the colonies that they formed as white, circular, convex, and translucent. Individually, they're described as, quote, non-motile short rods. The Aquahabitans phylum is known for its presence in the soil cycle. It's actually so important that without this bacteria, agriculture as we know it would be completely impossible. Their job in the cycle is to break down organic matter into nutrients, which can then saturate the soil, making it available to nearby plants for consumption. This behavior is super similar to fungi's role in the decomposition cycle. Fungi feed on organic tissue in order to aid, namely, decomposition. And this is so similar, in fact, that Aquahabitans were thought for a time to be a type of fungus. This misconception was so prevalent that it's even reflected in several of the naming conventions associated with the more major groups in this phylum. They are so important to the life cycle of both soil and plants that some members of the phylum even form symbiotic relationships with plants. In this relationship, they perform something called nitrogen fixation, and in exchange, the plant provides them with saccharides, essentially sugar. Actinobacteria also have a lot of other uses for humans, not just plants. Not only are they incredibly important to the soil cycle for the environment, but humans have also been able to use certain strains of actinobacteria for important agricultural developments, such as like insecticides, fungicides, and even things that promote growth. They are also well known for facilitating the advancement of medicines like antibiotics and antifungals. So they can be used both to inhibit and to exponentially increase the rate of things like fungi and plants. Something that I find really cool about this phylum is that the oldest living organisms on Earth are believed to be Aquahabitans bacteria, which survived for many, many years in sheets of permafrost. This fascinates me because according to certain studies, the biggest piece of evidence for the presence of this bacteria in the environment is CO2 being released from permafrost samples. This is strange to think about, not only because it suggests that these bacteria are at least somewhat operational after so many years, but also that they've somehow survived in such cold temperatures despite the fact that they can usually only survive in warm climates. Another cool fact about this particular strain of bacteria is the fact that its discovery is relatively recent. In a study published in 2007, a unique strain of bacterial DNA was isolated in the Daejeon Reservoir in central South Korea. With further research, it was ascertained that this DNA could be accredited to an entirely new strain of Aquahabitan, which was then named after the reservoir it was discovered in, hence Aquahabitans deichengensis. Thank you so much for your attention. I will pass the torch on to our next speaker.
My name is Emily Ramirez Miranda, and the bacteria I will be presenting is the Sigmonas hinkenensis. The environment where this bacteria was collected was in a wastewater treatment plant. Scientists in South Korea isolated this bacteria from a wastewater plant in Daejeon, South Cheongcheon province. The temperature in which this bacteria grows is between 4 to 37 degrees Celsius, or converted into Fahrenheit, it would be 39.2 between 98.6 degrees Fahrenheit. Its best growth temperature is 30 degrees Celsius, or 86 degrees Fahrenheit. The pH of this bacteria is between 5 to 10 on a pH scale, but it can either be acidic or basic. This bacteria does contain salt between 0 to 1%, and as mentioned before from one of my group mates, this salt is available in our households since it is table salt. There are a few things that make this bacteria special. It's a gram-negative, rod-shaped, and non-motile bacteria. Its oxygen tolerance is aerobe, and for enzyme type, it's cytochrome oxidase, which the activity is positive. Now moving on to the bacteria-soil relationship, Pseudomonas hinkenensis belongs to the Proteobacteria phylum, in which it pertains phylum bacteria of diverse properties and having similar gene sequences. Its role in the soil is to have the ability to metabolize toxic substances and to be able to survive with few carbon sources that are used as a nutrient. To conclude my part of the presentation, scientists in South Korea, isolated this bacteria from a wastewater plant in Daejeon. In addition to this, scientists continued their research and found this bacteria in Saudi Arabian soil as well. I now introduce you to the next and final presenter.
Hi, my name is Keili, and I'm going to be talking about Lacunasphaera limnophila. The environment that this bacteria is grown is in duck ponds in Germany, specifically in Wolfenbüttel, Germany, and the temperature that this bacteria grows the best at is 28 degrees Celsius, which converted to Fahrenheit is 82.4 degrees. The pH of this bacteria is 7.5, meaning that this bacteria is slightly basic. This bacteria does contain salt, and the salt that's in it is mineral salt. This is inorganic salts that need to be ingested or absorbed by living organisms for healthy growth and maintenance. Some special things about this bacteria is that it is mesophilic, meaning that it is an organism that grows best in moderate temperature, meaning neither too hot nor too cold, with temperatures ranging from 20 to 45 degrees Celsius or 68 to 113 degrees Fahrenheit. Another special thing about this bacteria is that it is also aquatic, meaning that it's suitable for a pond and wet environments. This bacteria falls under a group called Verrucomicrobia. Verrucomicrobia is a phylum of bacteria. This is a species isolated in freshwater, marine, and soil environments and are found to be more abundant during the spring season. There's evidence to suggest that Verrucomicrobia are abundant in the environment and important for soil cultures. They are found everywhere in the soil but only at low frequencies. This species has a role in soil fertility and also has the ability to adapt to the changing ecosystem. The Verrucomicrobia phylum can be distinguished from other phyla within the PVC group by the presence of several conserved signature indels, or CSI. Scientists isolated this bacteria from soil samples from a duck pond in Wolfenbüttel, Straßgraben, which is located in northern Germany. This bacteria is a fairly new phenomenon because it was all documented in 2017. Further studies revolving around the general phylum group of Verrucomicrobia have been in effect revolving around soil fertility.